Hi, I'm Adam Coscia, a PhD student at Georgia Tech. On behalf of my amazing collaborators at Georgia Tech, Caltech, Art Center College of Design, the Monterey Bay Aquarium Research Institute, and NASA's Jet Propulsion Laboratory, I'm here to present our work on DeepSee, an interactive workspace for studying deep ocean ecosystems. In this design study, we established a multidisciplinary collaboration with scientists at Caltech that are advancing our understanding of the deep ocean. The deep ocean is home to rich, complex microbial ecosystems that facilitate key environmental processes, including the transformation of greenhouse gases like methane and carbon dioxide. To study these fascinating environments, scientists first need to collect sediment samples during research expeditions, where they use autonomous and remotely operated vehicles to map, explore, and collect sediment samples from the ocean floor and bring the samples back to the ship for downstream processing. However, fieldwork is a time-consuming and expensive task that usually results in a very limited number of samples. Seeing prior sampling history to decide where to sample next can help maximize time spent conducting fieldwork, as well as increase the scientific return or long-term value and usability of prior sample data in the lab. To address this gap, we accomplished three primary goals in this design study. First, we designed a visualization system that can enable scientists to explore spatial trends between samples in context of the environment. Second, we studied how scientists' existing workflows changed when deploying visualizations during a real field research expedition. Third, we reflected on broader design guidance for building future visual analytics systems around visualizing multidimensional fieldwork sample data. To decide where to dive next, scientists analyzed sample data collected during prior fieldwork expeditions logged in Excel tables, alongside separate, high-resolution images and maps of the seafloor. This format requires scientists to manually correlate processes between image and tabular data types, while keeping an accurate mental model. The tables and maps described real-life counterparts, cylindrical cores of sediment, rock, and water collected from the seafloor. Thus, we aim to visualize the distributions of geochemical and biological parameters in each core in multiple dimensions as a function of latitude, longitude, depth under the seafloor, as well as over different time periods, in a single cohesive interface featuring both table and map data. Our solution, DeepSee, is a web browser-based workspace that presents interactive side-by-side views of 2D geological and biological landscape maps, as well as 2D visualizations and 3D interpolations of physical, geochemical, and biological parameter gradients in deep sea sediment cores. DeepSee combines three different coordinated views in a single interface. The first view is the map view, which plots sample data from the Excel tables on top of maps of the seafloor. By combining tabular and image data, users gain more context about spatial trends in the data from previous expeditions that were previously hard to identify. Our designs facilitate the process of correlating image features with tabular data, such as where prior samples align with surface bacterial expressions or geological formations on the seafloor. They also help scientists interactively identify a subset of cores for further analysis in other views. Here, users can filter cores from the map by core features, as well as between different cruises, click and drag to interactively zoom in and out of the map, hover on the map, revealing details about previously sampled cores on demand, switch to a different map while preserving the existing viewport to maintain context as users zoom in and out, draw and take notes on the maps using annotation tools, which persist as you switch map layers, allowing users to see how biological features correlate with seafloor geography. And finally, select a subset of cores to conduct further analysis in the following two views. The second view is the core view, which arranges selected cores side by side and visualizes their biogeochemical parameters as horizontal bar charts. By aligning bars according to the depth horizon, scientists can explore the distributions of subsurface parameter gradients between cores, such as where sulfate might correlate with bacterial expressions. The third view is the interpolation view, which visualizes the same core parameter distributions in 3D by interpolating missing values in between samples and rendering those values as space-filling volumetric pixels in an interactive, navigable 3D space. With the interpolation view, scientists can gain intuitions about how different interpolation methods lead to different predicted subsurface gradients, such as how sulfide might be diffusing around deep-sea thermal vents. We found that our collaborators were more familiar reasoning about complex ecological processes in 3D. Interpolations are run in real time, enabling scientists to perform new predictive analyses and ask questions about what parameter values might look like in unsampled locations, a previously difficult task to mentally model and evaluate. We provide two interpolation methods that can be switched on the fly: linear interpolation and space-filling natural neighbors interpolation. Scientists can also adjust the grid sizes on the fly. We also leveraged uncertainty color palettes, which visually suppress the color of interpolated parameters which are less certain in the data. This provided a huge benefit to scientists in quickly identifying where their data was stronger and where future sampling could increase confidence in the interpolations. We provide several reconfiguration interactions to help users navigate the view, including changing the depth axis and changing voxels to cylinders to match the shape of real-life cores. To see patterns inside the filled space, scientists can clip through their data by parameter values, as well as by latitude, longitude, and depth. And finally, we provide a selection tool to download interpolated data in the same format as the input core data, allowing scientists to compare predictions with later samples and update their hypotheses. Working alongside our collaborators, we explored how DeepSee supports several usage scenarios. For example, scientists can use DeepSee to plan a follow-up cruise to collect cores at a previously visited site. By correlating the presence of two areas with high abundance of bacteria in the core view on the right, with expressions of white microbial mats in the map view on the left, DeepSee helps maximize the value of limited samples to help decide where to sample in the future. DeepSee can also be used by API during a cruise to quickly select new sampling sites. Using the annotations in the map view on the left, the PI can synthesize a change in plans and communicate to submersible drivers if they discover the map doesn't match what they're currently seeing through the cameras of the submersible. They can further use the interpolation view to quickly generate a predicted subsurface gradient in a new location that looks promising for sampling. Having the ability to visualize unseen gradients with limited data in the field can help researchers make smarter, more data-driven decisions. To evaluate our designs, our collaborators deployed DeepSee on an actual research expedition in October 2021 to sample cores around vents and seeps of the Pescadero Basin in the Gulf of California. I want to thank Victoria Orphan and the Orphan Lab at Caltech for taking a leap of faith and testing our prototype live on a ship out in the ocean. Before embarking, we found DeepSee helped the most during pre-cruise planning by getting everyone up to speed on site conditions, prior data, and performing preliminary investigations of the data for correlations and patterns worth exploring once embarked. During the cruise, we observed that DeepSee was mostly used for matching prior sample data to the current dive location and tracking conditions to assist the lead researcher in decision making. After the deployment, we conducted interviews with the same experts to understand how DeepSee supported the scientists' research, workflow, and teamwork. We found that during pre-cruise planning, DeepSee helped researchers discover holes in their sampling strategy, such as where bacteria are abundant at the edges of sampling sites, by enabling fluid interaction between the microscale individual cores and macroscale collections of cores across an entire map. We also found that new workflows were developed where scientists would actually revisit previously analyzed data to see if new insights would appear, increasing the scientific return of samples. And finally, the intuitive modular design of DeepSee with individuals' views side by side, supported diverse goals within the team, including pre-selecting sites, interpolating data, aligning logbooks from prior expeditions, and reducing the need for multiple complex software programs running during the cruises. With our findings, we synthesized several implications for the design of future visual analytics systems. We observed time pressures when tracking data added on the fly during the expedition, which we did not prioritize as a user task up front. So, one solution to overcome this could be taking a mixed initiative approach, where the system foregrounds specific cores or interpolations automatically based on pre-cruise interactions with DeepSee. Use of 3D views, context-aware visualizations, and direct manipulation helped scientists continue thinking and communicating in terms of their science rather than requiring context switching, particularly in time-sensitive environments such as during a cruise. By overlaying tabular and map data at multiple scales, scientists could ask new questions of their data and utilize old data in new workflows to gain additional insights. Interactivity also played a key role in encouraging scientists to explicitly model their assumptions about the data and test hypotheses on the fly, leading to increased interest in the fieldwork community for more live, on-the-fly visualization tools. Finally, our study also revealed several avenues for future work. Our scientists were interested in additional capabilities for advanced microbial data analysis in the core view, as well as more sophisticated models in the interpolation view. We're personally interested in exploring how computational notebooks like Jupyter might extend the longevity, reach, and usability of our visualizations in other fieldwork-driven communities. And there's also interest in capturing the interactions that operators have with DeepSee to inform the design of future autonomous sampling systems. By visualizing samples in the context of the environment, bringing together multiple data types in a single interface, and providing capabilities to predict values in unsampled locations, DeepSee offers a new approach to visualizing 3D point samples in fieldwork-driven science. DeepSee is live and open source, so please check it out. Thank you.